Welcome back. We left off with the concept of lines of cleavage. From here on out, it's just kind of clean up. There's only a couple of more key concepts, a little bit more vocab. We've already passed the hardest part. Those layers were probably the hardest part. So again, nerve fibers in the dermis. We're going to have tactile receptors. We're going to have blood flow, and we're going to have glands. So specifically sweat glands, exocrine glands, which secrete products into ducts. Those blood vessels within the dermis can vasoconstrict and vasodilate. So for example, if your body temperature was low, that would send a signal to the hypothalamus that the temperature is too low, and that hypothalamus would utilize some effectors such as smooth muscle around your blood vessels, and that would cause vasoconstriction, and that vasoconstriction would help shunt that blood supply, which carries your heat, to the internal environment and preserve it. In the opposite situation, you would have vasodilation. Smooth muscle around those blood vessels would release and allow for more blood flow close to the skin surface, which allows you to radiate heat out to the environment. Obviously, that's not the only scenario where you would get vasoconstriction or vasodilation. That is just the temperature control aspect of it. Anything from blushing to, again, so hemostasis, we have vasoconstriction if a blood vessel is broken. A number of scenarios lead to these situations. Again, that subcutaneous layer is not part of the integument. It stores energy, thermal insulation, protection. Adipose is really pretty good at being protective. You have adipose in a variety of interesting places, like around your eye, in your orbit. Your eye is padded out with adipose tissue just to keep it in place. Okay, I think we're good there. Some other structures derived from the epidermis, your nails, your hair, and then we're going to go into details about glands, and there's a little bit more vocab associated with each of these, but not too much. Vocab associated with your nails includes the nail plate. I think that one's pretty obvious. The nail, let's see, root is going to be back under here. You've got a hyponychium and an eponychium. So eponychium is the surface. Hyponychium is going to be this edge of the free nail, so that sort of paler area of your nail underneath the free edge, that's going to be your hyponychium, and it's spelled N-Y-C-H-I-U-M, nychium. That eponychium, let's see, do we even have it on here? Is going to actually just be that cuticle portion. So if you get your nails done, they might push back that eponychium, the cuticle. Nail body up here. What else am I missing? The nail.
Actually, the hyponychium is the nail bed.
So no, the nail bed is going to be, if we zoomed way in here, thanks for the points of clarification, I am kind of rushing this. The nail bed, if you zoom in here, you see this sort of foldy fold. That's your basement membrane for your epidermis, and remember that this nail bed is going to be derived from your epidermis. This is very, very modified keratinocytes coming out. The nail bed is going to be the cells immediately below the nail plate. The nail plate are those modified keratinocytes. They're being formed at the nail matrix. Eponychium is cuticle. Hyponychium is just below that free edge. If you get French tips, they're putting that white nail polish on the surface of the hyponychium. And I know that applies to everybody equally in this room. Everybody knows all about French tips. Yep. Yeah.
Get them once a week.
Once a week, minimum. Okay, any questions about that? Thank you for asking me to clarify those. Okay, so the nail matrix are going to be those cells that are dividing, those keratinocytes that are dividing and modifying themselves into that nail plate form. And this is literally the only image you have for fingernails for lab practical purposes. That's it. That's all I can test you from. With hair, what's that? With hair, again, you've only got the one slide for me to test you for images for hair. It's going to be very similar. I mentioned earlier that you've got a basement membrane that invaginates into the dermis so that you can have those stratum basale cells undergoing mitosis and once again creating modified keratinocytes that lack a nucleus, that are deceased, but in a very structurally strong way, very, very keratin rich, that's going to form down here in the hair bulb. So this whole thing is the bulb. You do have some layers here and some vocab words that I can hold you to as per our vocab list for the integumentary lab. The hair matrix, very much like with your nail matrix, some of these cells are dividing and forming here that are going to make the shaft of the hair. An internal root sheath and external root sheath. The hair papilla is also going to include some not nerves, but arteries and veins. And then you have the hair follicle, which is going to be this outer layer, and the shaft itself, the hair shaft, coming up here into the external environment. So that's all vocab I can hold you to. And in general, all you're seeing is that the cells are being synthesized and being added from way down here. And then by the time it's out here, you can already see these cells are dead. They have no nuclei. These are entirely deceased. So if you just popped some keratin on top of this shaft right here, do you think that's really going to do anything? Probably not. It's not going to absorb into much of anything. Okay, adhered to, yes, question.
You may have said this already, sorry.
That's right.
So on the hair, like as far as what I have to go, is it just pain or anything that you would hold us accountable for?
For which one?
Hair.
For hair for what?
The black practical part of the pictures. Would you show us A and B and expect us to point out stuff on?
You mean like this picture than this picture?
Yeah.
Yes, I could use either of these images to test you on.
Okay.
Absolutely. It's a good question. Attached to every hair shaft is going to be a bit of smooth muscle, and this is where those histology skills really come in handy, recognizing that little tiny clump right there as smooth muscle. That's the erector pili muscle. So we are not into muscles unit yet. This is the only muscle I can hold you to right now. It's smooth muscle, so it's involuntary. You have no conscious control. You can't flex your arm hairs no matter how bad you try. But in response to cold or other unexpected stimuli, this could contract and it's going to straighten this hair shaft out. And that causes a sort of it actually is relatively effective on humans. This has been tested for maintaining body heat. We thought, when we became human, we lost some of our body hair compared to other primates, and maybe it's not as effective as an insulator, but it actually does hold just a layer of heat close to our body, even if you've got, my arm hair situation, which is very, very pale and useless looking. Okay, so that's pretty cool. Its hair is protective. It helps you retain heat, sensory reception with your root. Hair roots do have specialized sensory nerve endings known as root hair plexuses, which is going to be why it hurts so much to pluck that out. But they're so sensitive that even if you nudge that hair shaft, that they're going to be able to feel it there. Visual identification, chemical signal dispersal, especially armpits and groins. It is very controversial if we have pheromones or not. I'm of the opinion that we do. The science for this is terrible. So saying chemical signal dispersal means that we have pheromones, and that's part of the job of hair, and I happen to agree with that. You're never going to find a scientist that says we definitely have pheromones and they're definitely acting on us. And if we want to talk about that at some point, we can. Probably not on a YouTube scenario where you're listening to this repeatedly, though, because it's off topic. Yes?
One more question.
Yeah.
So when they do laser hair removal and target, they say the hair follicles and they
Yep.
say they kill it, which part would that be? Like the hair bulb
It's a good question. So different hair removal treatments, if you're plucking it, then your hair papilla is still going to have to be alive for that root to come back, right? If you're getting electrolysis, I think is the right word for it, that's actually going to kill off these cells so that it can't come back. Laser hair removal sometimes manages to kill this and sometimes doesn't. So it's usually you can't get away with just doing one laser treatment, as I understand it. I'm not an expert in this field.
6 to 10 or something, but they say it can be permanent, so I didn't know
Yeah.
if it does.
It's going to be dependent on, is that laser actually going to successfully come down to that papilla, to those basal cells that are undergoing mitosis and synthesizing those keratinocytes.
So it would have to go all throughout.
It would have to get all the way down from to here. If you're not killing down here in the bulb, then it's going to come back. Yeah, you can also kind of see the source for ingrown hairs here too, right? If you pluck that out and then it grows, but let's say this the space for that hair shaft is obscured in some way via like a healing process, then it's still going to grow. It's just not going to be able to get through all of these layers very easily, and that would be an ingrown hair. Okay, double check that your PowerPoint has this formatting correctly. On an older version of this, this line used to be connected to the concept of alopecia, which they are separate concepts entirely. Alopecia is a pathology in which people start losing their hair permanently, and I believe it is autoimmune. The immune system attacks the hair follicles, and they die permanently.
It says autoimmune next to alopecia on here.
Okay, so I have updated those. This is an older version. So that's an autoimmune condition. It is permanent. That is a separate concept from hair thinning in both sexes after the age of 40. That is just universally true. Both men and women start to lose hair, decreased vascular supply just as we age in general. That's about it. It's nutritional, it's age-based, less mitosis, so less hair growing. That is different from true or frank baldness, which is male pattern baldness. So if you know any men who are losing their hair, you can tell them they are just too manly because it's actually moderated by testosterone. It means that the follicles are responding to that testosterone in such a way that they start to fall out, which is weird, but yeah, those men are just too manly. Their hair can't handle it. They leave. That's how you do that. The treatments, we've got Rogaine, we've got Propecia. Pregnant people should not even touch Propecia. It's a bad thing. Okay, a little bit more vocab, a little bit more reasoning at the cellular level coming right up. You've got two major types of glands, one of which has two subtypes. Sudiferous glands, or sweat glands, produce watery solutions, and you have more than one kind of sweat. You have stinky sweat, and you have non-stinky sweat. And that's literally the difference. I'm not exaggerating about that. And then you also have sebaceous glands, usually associated with your hair follicles, not entirely associated. So that oily, like hair oil, skin oil, is really coming from sebum, and that's going to be a separate substance from sweat. Just because your hair is oily doesn't mean your head is sweaty. Those are separate concepts. The major difference between Merocrine sweat and Apocrine sweat is how those cellular products are getting into the exocrine glands. It has a really major impact on the consistency, the smell, the environment that they create. So Merocrine sweat glands, they're actually using exocytosis, just kind of a normal cellular process. They make something that's really mostly just water. So the palms of your hand, if your palms get sweaty, you never sniff your palm and think, ugh, I smell so bad, I'm so sweaty. That's not really a thing. That sweat is Merocrine sweat. It's not going to smell especially bad. It's mostly water. Apocrine sweat gland is different. This cellular product is actually pinched off chunks of cells, which means that this is going to be a really, really great environment for bacteria. It's got all of those nutrients living within the cell to feed on. That's going to be what causes the odor, the bacterial environment caused by pinched off bits of cell that have been secreted. So these are the ones that are found in your axillae, which you now know for sure is your armpit, especially in the groin region, the anal region, in the areolar region of nipples as well, although personally I've never sniffed nipples and had them smell weird, so whatever. You know you've done it. All right, so mnemonic devices for that, I had one cohort say, Apocrine sweat glands make you smell like an ape. If it works for you, awesome. If not, we'll figure something out. So Americrine versus Apocrine, both of this being sudiferous or sweat glands. Last gland type is sebaceous gland, and they secrete sebum. If you don't wash your hair for a few days, that buildup is sebum. Now this actually increases oil production during puberty in response to some of those sex steroid hormones. And there's some histology to go along with that. I generally don't hold you to the histology of these glands. But you do have representations of them in our general integumentary image. So I could point to that and have you recognize that that is a sweat gland versus that's a sebaceous gland.
Which one's a sweat gland?
This is the sweat gland, so it's really coiled, and it comes out to the surface of the skin. Whereas the sebaceous gland is secreting, in this case, onto that hair follicle.
So then can you use sweat gland, or you have to use like the...
Sweat gland, I would say, would be acceptable. Yeah. Okay. Skin cancer. Now that you know some of the layers of the skin and some of the cells of the skin, your major types of skin cancer are going to make a little bit more sense. So for example, if we're talking about a basal cell carcinoma, what layer do you think that came from? What strata? The bottom, stratum basale. So this is going to be the deepest layer. And then squamous cell, what's the description of a squamous cell? How would you define that? Kind of flattened, so it's not going to be a basal cell, it's going to be a cell that's farther away from that stratum basale, closer to the stratum corneum. So this might be in the spinosum layer. And then a melanoma, what cell type would you expect that to be of? Melanocytes, exactly. So here's how each of those can look, and these are specifically cancers. There are benign forms that each of these cell types can undergo instead of cancerous forms. So melanoma is the least common, but it's the most deadly. By contrast, basal cell carcinoma is the most common and statistically least deadly, as I understand it. We're going to do a little ABCDE. It's not something I would hold you to for the exam, but I think it's clinically important enough that everybody should know it off the top of their heads at all times. Now this is technically ABCDE for melanoma, but I think it works for a variety of different types of skin cancer. Has anybody ever seen this one before? What's A? Asymmetrical. If a mole is asymmetrical, you may want to get it checked. B is for border. Is the mole clearly defined? Look at this guy right here. Is that a really clearly defined border, or are you going to have a hard time saying precisely where that mole starts and stops? It's a relatively poorly defined border. And you can see it here as well. It's a relatively poorly defined border. Okay, C is for color. For melanoma, that's actually pretty obvious. If you've got a very large patch of very darkly stained skin compared to the surrounding area, then that's a warning sign. D is for diameter, which is going to be about what? Size, yes. So if it's, I think the rule of thumb is wider than the eraser on a pencil is when you start worrying about it. E is for, there's a couple of options for E. Evolving is one of them. And evolving may be the most important out of any of these. If it's changing over time, that means cellular growth is occurring. one way or another. So if it's evolving over time, that's your #1 red flag for any type of mole that you have. What could be the other E? Evolving, evolution, elevation. You can actually see each of these. That squamous cell carcinoma is elevated, that basal cell carcinoma has an elevated area. Some of these can cause pitting as well, so any change in elevation would also be very worrying. They can cause ulcerations as well. And that melanoma is also elevated. So sometimes people say change for C as well, meaning the same thing as evolving under E. Okay. I mentioned earlier one of the biggest threats for burns is actually fluid loss. It's not the only danger, but it's a big one. In addition to killing cells, it does something called denaturing proteins. So the structure of proteins is that they are folded. Denaturing means they lose their folds and they lose their shape, so they lose their function. And when your proteins are denatured, the machinery of your cells can't work anymore. It's basically you've melted it. It's just a way to say that. But again, that immediate threat, dehydration and electrolyte imbalances are very important. All of those free proteins can lead to renal shutdown as well. So emergency departments have the rule of 9s. If you're not a math person, 4.5 is exactly half of 9, and 18 is exactly 2 times 9. It's okay, not everybody's a math person. And we're going to specify that the front of the leg is 9, also the back of the leg is 9, meaning that this leg is going to be a total of 18. The anterior trunk will be 18, the posterior trunk will also be 18. And I think the number is 23%. Do you know off the top of your head? For, there's this, oh wait, no, I have it. Critical if more than 25% of the body has 2nd-degree burns, or if more than 10% of the body has 3rd-degree burns. So that's why we use the rule of 9s, is to quickly get to that percentage of, if you have your entire anterior trunk with 3rd-degree burns, you're already critical. That is an absolute emergency.
For EMS is 25 and 10.
Yeah, just 25 and 10. Okay, cool. If you work in a burn ward, there are more specific guidelines than this. This is, I think, general emergency department. Difference between 1st, 2nd degree. 1st-degree burn is just going to have some redness, some swelling. It's going to hurt. A sunburn, for most people, is going to be a 1st-degree burn. Personally, I've had 2nd-degree burns as sunburns, but that's because of this situation right here. Yeah, somebody else. So it is possible to get a sunburn for 2nd degree. For the most part, I do, jazz hands, want you to know that most sunburns would be categorized as 1st-degree burns. 2nd-degree burns mean that we've gotten down to the dermal layer. That superficial burn was just the epidermis. It did not get down to the dermal layer. 2nd-degree burn gets down to the dermal layer. And just as a side note for now, that dermal layer is relatively thick. There are 2 degrees of 2nd-degree burns. You can have a superficial or a deep 2nd-degree burn, depending on how deep through the dermis you got. If you get down to the hypodermis, we'll call that a full thickness or 3rd-degree burn. Now, where are most of your nerves? Affected by 2nd-degree burns is correct. In the dermis. Most of your nerve endings are going to be in the dermis. You do have some for the epidermis, which is why that superficial one, that sunburn, is so painful. Partial thickness 2nd degree will also be painful because you are exposing but not destroying all of those nerves, right? Is a 3rd-degree burn going to necessarily be painful? Nope, not in the zone of necrosis. Where it's completely dead, you've completely eaten through those nerve endings. They are no longer sending pain signals. So this area is going to be painless. Over at the edges, where you have some zones of swelling, zone of erythema, that will be painful, but the body of that burn will not be painful. For some of these PowerPoints, I have links as well, so those may be worth checking out if you want to go into more detail about any of that. Okay, skin grafting may be necessary for third-degree burns, or usually necessary. May also be worth noting that second-degree burns are characterized by blisters. First-degree burns will not have blistering. Okay, stages of wound repair. What you're going to see is any broken blood vessels creating that hematoma, that collection of blood. You're going to see mast cells, a modified white blood cell, go into the region and trigger inflammation. We're just going to say inflammation for now. We're not doing a ton of detail on this. Gradually, more white blood cells are going to come in. We're going to have some phagocytes, some eating cells, come in and remove debris. We're going to have a lot of clotting around the edges, isolating the area, a lot of migration of cells that are there to help you out. Fibroblasts. What do we mean by fibroblasts? Fibroblast. Building fibers. Perfect. Well done. Eventually, we're going to restore that stratum basale, and those cells can undergo mitosis. Those fibroblasts are going to put down new collagen in the area beneath the wound. Eventually, we'll slough off that scab, and that scar tissue will remain in the dermal layer. Some people, especially with specific genetic backgrounds, will be more prone to keloid scarring. And that's going to be overproduction of collagen at this time, with some interesting genetic determinants for that. Not 100% figured out why that happens in some individuals and not others. And that is it. Any questions before I stop the recording? All right.